Today we're going to be talking about various social behaviors and the roles of two hormones in contributing to how those social behaviors are manifested. We're going to be talking about oxytocin and vasopressin. So first we got to define social behaviors. One definition is that it encompasses a wide swath of behaviors where individuals interact with others or where individuals are required to interpret the actions of others. There's a lot that we can call social behaviors, and just about any sort of interaction between two members of the same species can be referred to social behaviors. Some of the obvious ones are maternal-paternal behaviors, just general social interaction or exploration with the same sex or opposite sex. So this is sort of a sex-independent definition. Courtship and reproduction, by definition, naturally assumes that opposite sex interaction is there for most species. Then there's prosocial behaviors. So we get into a little bit more specific about the type of interaction, the nature of the interaction. So prosocial behaviors such as altruistic behaviors. To be altruistic means to help others at some consequence for yourself or some cost to yourself, helping out others. Aggression, like these guys, they're fighting. You can see they're not too happy. Many species will show aggressive behaviors. Virtually all of them do at some point. And so this is one type. And then of course, play behavior is something that's growing to be more and more appreciated as an important aspect of how animals learn to interact with each other. And so there's a fair amount of research happening on this as well. Today, we're mostly going to be talking about maternal behavior, since there's been a ton of research on the underlying neurobiology of that, as most of it coming from rodents. We will be discussing a little bit of aggressive behavior, and there'll be some points where we would touch on a few other behaviors as well. So this is the portion of the hypothalamus that involves hormones being released from the posterior lobe. So we're going to focus on that right now. So up to this point, we've talked about the anterior lobe of the pituitary, the adenohypophysis. Now we're going to be talking about this neurohypophysis. So the neurohypophysis, as I said, it's called the neurohypophysis because it doesn't have any secretory cells. The secretions in the neurohypophysis come from the axons of neurons themselves, and it is filled with axons from a cell type called magnocellular cells. So these aren't parvocellular; these are magnocellular. They're called magnocellular because these cells are very, very big. The hormones that are released from the posterior pituitary are oxytocin and vasopressin. And they get released into a capillary bed that goes into the neurohypophysis, and it gets released directly into that capillary bed and then sent out to the bloodstream directly. So there's no middleman exchange in the median eminence / infundibulum. There's no transfer, and there's no secretagogue here. It is just neurons releasing these factors, oxytocin and vasopressin, directly into the bloodstream in the posterior pituitary. So a pretty straight shot. Oxytocin is in the media referred to as the love hormone. It's a lot more complicated than that, as we'll learn. But it is very important for social bonding and maternal behavior. And then there's vasopressin. Vasopressin, this is a multifunctional hormone. It gets its name, vasopressin, because it's an antidiuretic. It regulates blood volume and salt concentration. So it changes the pressure in the vasculature, hence its name. Cells that make vasopressin, they sense changes in blood tonicity, and then they can release more vasopressin upon water loss. So this is something that vasopressin is very important in, but it turns out that it's also involved in the regulation of various kinds of behaviors. And in particular, we're going to talk about its relationship to aggressive behavior. So where are magnocellular cells located? We can identify location, obviously, through doing basic immunostaining procedures. And so what we've done here is that this is a coronal section of a rodent hypothalamus. Ventral is down here, dorsal is here. This is the ventricle here, the third ventricle, this little space here. And this location here is the paraventricular nucleus. You can see this is the ventricle, and this is running right along the ventricle, sort of more on the dorsal side of the hypothalamus. So there are other brain areas here that are filled with neurons, but we can't see them because they don't have oxytocin, which is shown in red, or vasopressin, which is shown in green. So we see that that red cells and green cells are largely located in the same nucleus, in the paraventricular nucleus. The other thing that you should see is that there's virtually no yellow. So what that means is that cells either produce oxytocin or vasopressin. They don't produce both. And so we have even though they're all located and jumbled up in essentially the same brain area, there's some subtle differences, with red being a little bit more medial and the vasopressin being a little bit more lateral. In general, in the paraventricular nucleus, they're just mostly located in the same area, but not in the same neurons. So we're going to talk about oxytocin a bit, and its various roles. One major action of oxytocin, something that's very well understood, and its role in the periphery is to induce uterine contraction of the uterus in order to start labor and speed up labor. So there's a specific neuroendocrine mechanism that's involved with this. When it's time, when the baby's ready to come out and birth is about to happen, if the baby's oriented in the correct orientation, the head of the fetus will be pushing up against the cervix. So that's sort of the first step here. And so the baby's pushing up there, and baby's ready to come out. This initiates nerve impulses. So there's all kinds of touch and stretch receptors down here in the cervix. This information gets transmitted up to the brain. This pressure stimulates the pituitary gland to secrete oxytocin. So those oxytocin positive neurons in the paraventricular nucleus are stimulated to release oxytocin. So the amount of oxytocin increases. The oxytocin is then carried to the bloodstream to the uterus. And then oxytocin acts on oxytocin receptors in the muscles within the uterus, stimulating uterine contractions, pushes the fetus forward towards the cervix. So then what happens? Well, we have now even more pressure on the cervix, which is going to induce even more nerve pulses from the cervix to the brain, causing more oxytocin to be released, which gets carried into the bloodstream, inducing even more contraction. You can see that this is a classic positive feedback loop. Oxytocin is inducing contraction, which is causing more pressure here, which induces more oxytocin. And hence, it becomes a very sort of painful process because of this positive feedback loop with virtually no cognitive control from the mother. One thing about therapy is that sometimes labor can be somewhat sluggish. It doesn't seem like it's proceeding very quickly. The contractions aren't happening as frequently as they should, or they're not as strong. There are synthetic oxytocins, one called Pitocin, which stimulates and expedites labor. There's, just to provide a little bit of additional context, there's a fair amount of debate over the use of Pitocin that in some circumstances it may actually exacerbate problems in delivery. But in some cases it's definitely important. So that's as far as oxytocin and its very clear critical role in uterine contractions. But it plays a role in all kinds of bonding behavior, including bonding between the mother and the offspring. So just like anything that you can measure, especially something complex like mothering behavior, you can find variation. Not all mothers are the same. Some mothers are good mothers, and if we're talking about a good mother rat, she will lick and groom her pups, licking and grooming, or LG behavior. She will give them extra space to suckle underneath her underside, so she will turn to her side and let them nurse for long periods of time. If you take a pup away and it starts to squeal a little bit, she will then go and retrieve the pup and bring it back with her, and she will spend a lot of time with her pups. We can measure all of these things, and in general, you will see that there are mothers who are very good mothers, and they spend a lot of time with their pups. There are also bad mothers. So a bad mother hardly ever licks and grooms her pups. She hardly ever spends any time with them, so she's not spending she's not giving them a whole lot of tactile stimulation, and she takes a very long time to retrieve pups that have been pulled away from her. She's a bad mother. So we can see this variation in rats and mice, and we can measure what the heck is going on in the brain and what factors might be involved with this. So we're going to talk about a bunch of experiments that are giving us a hint of why some mothers act good and why some mothers act bad. So in addition to the positive feedback loop that oxytocin induces as far as uterine contraction is concerned, there's also some positive feedback loops that oxytocin is involved in with stimulating maternal behavior, as well as just overall pair bonding between parent and offspring. So the initial reaction of giving birth, being pregnant and having the birth happening, that's called partum, that you have this huge surge in oxytocin inside the mother. This primes the mother to be stimulated to induce maternal care and induce social bonding between parent and offspring. Then there's a series of behaviors that reinforce this surge in oxytocin. So with this high oxytocin levels, stimulating maternal care as the first step, the mother then engage in parental care of the offspring. This parental care of licking, grooming, close touch, tactile stimulation, feeding, responding to calls of distress, this increases oxytocin levels in the pup. The pup then shows affiliative attachment behaviors to the mother, and this ends up creating a bond. This touching and nursing behavior induces stimulation of oxytocin to within the mother. So now there's more and more oxytocin being released in the mother, which stimulates more parental behavior, and you end up with this positive feedback loop that occurs. So this then can lead to them to display high levels of parental care when they really reach adulthood. So this positive feedback loop of having a high licking and grooming experience as a pup will lead to the next generation pup, the mothers, when the daughters become mothers themselves, they will be more likely to show high parental care, a lot of licking and grooming in the next generation, which then stimulates this positive feedback loop of oxytocin, inducing more licking and grooming at the next time. So we can see positive feedback loops happening within an individual, across individuals, within one generation, and then within even across generations. So multiple positive feedback loops. So how does oxytocin shape maternal behavior? And more broadly, just social bonding. The several forms of sensory stimuli can trigger oxytocin release. So clearly, this social interaction involves the sensations. There are all kinds of things that are happening within that the animal is experiencing that would facilitate this bonding experience. So for the mother, there are a variety of stimuli. There are physical stimuli, such as nipple sucking, stroking, uterine stimulation that will induce this surge in oxytocin release. This leads to certain physiological outputs. We know that oxytocin is actually very important for milk production, another positive feedback loop. There are auditory stimuli, so infant vocalizations. This is true for humans, but also for rodents, that the little squeaky noise that rodents make, or baby cries for humans, That is an auditory stimuli. You don't even have to see the infant in that case, but that can trigger an oxytocin release as well. There are odors. Odors can figure into this as well. This is not as true for, say, humans, but it is true for a lot of other mammals, including rodents. In fact, we'll talk about this in a little bit more detail about the neural control of this. But smelling the baby can have this influence as well. Eye contact is very important for certain species. This is especially true in humans. Seeing babies itself can trigger an oxytocin release. Dogs and humans, they can have affiliative contact that actually can be influenced by oxytocin as well. That's just mediated by visual stimuli. So all of these factors will lead to, say, surges in oxytocin, and there'll be behavioral outputs such as maternal behavior, social bonding, social cognition behaviors, anxiety relief is another aspect of oxytocin release, a whole variety of factors. So how does it act then? Oxytocin is influencing all these different behaviors, so a huge range of behaviors. There are a lot of different areas of the brain that are sensitive to oxytocin. So just like any hormone, the hormone itself, in this case oxytocin, circulates around the brain and then binds to receptors that are located within the brain itself. So these are a series of sections, coronal sections through the rodent brain that have been processed for in situ hybridization. Again, the same technique that we talked about when we were looking at CRH in the paraventricular nucleus. So this is just simply showing where mRNA for oxytocin and receptor is located. So in this procedure, they did essentially the same thing, but then instead of looking for CRH, they're looking for oxytocin receptor. These are areas of the brain that will be sensitive to oxytocin. So this is in the olfactory bulb. You can see the dark areas, that shows where there's oxytocin receptor, and there's a lot of oxytocin located there. This is also true in various parts of the cortex. You can see many areas in the cortex of oxytocin receptor. This is also true in the hippocampus. So a bunch of areas in the brain have oxytocin receptor, which means when oxytocin is being released, it's surging and it's going to be binding to the receptors throughout the brain, inducing changes in behavior and leading to these changes in maternal behavior or social bonding. So it can have a huge effect through just the release of the hormone. So we're going to talk about what factors will influence maternal behavior. There are three that we're going to walk through. We have experience, sensory stimuli, and hormones, and their overall influence on maternal behavior. First, let's talk about experience. Previous motherhood affects maternal behavior. So this is the experience of having been a mother all of a sudden will lead to the likelihood of expressing maternal behavior. A couple of terms here. Nulliparous means never pregnant, and primiparous means having been pregnant once. So female rats that have never given birth show a high latency to display maternal behavior if given orphan rats. On the y-axis here, this is the number of days it takes for a female rat to be exposed to orphan rats and to prime them to all of a sudden start maternal behavior. So on the very first day, it takes a very long time. The likelihood of seeing maternal behavior is very, very low. On average, if you're talking about a relatively young, naive maternal rat, it takes about 6 days for them to show maternal behavior. If you follow the nulliparous line, you'll see all the way up until about 120 days or 130 days old. These rats, it gets a little bit better, but it still takes about 4 to 5 days of being exposed to infant rats in order for them to all of a sudden start showing maternal behavior. Once pregnant and given birth, the term primiparous indicating first pregnancy, a female rat will quickly display maternal behavior. So at this point, so the groups are the same, right? So because you can't get, they didn't get the rats pregnant very early on. At day 60, they split the rats into two groups, and that's when some of the rats were pregnant and some of the rats were not. You can see when pregnant, it still takes a long time for the primiparous rats to show maternal behavior. It's really no different than the rats that are not pregnant. But as soon as birth occurs, and just prior to birth, you can see all of a sudden there's a major switch in how long it takes for these rats to show maternal behavior towards infant rats. And once they've experienced during when they are raising their own rats, it takes no time at all for them to show maternal behavior to orphan rats. And once they have done taking care of their babies, lactation ends. It takes about 20-25 days for them to finish, raising the pups when they're mostly on their own. Now you can look even a month after having taken care of the rats. The mother rats who were mothers, when they're exposed to infant rats, it takes less than a day for them to show maternal behavior to orphan rats versus rats who have never had any birthing experience. So it looks like the priming of having been a mother makes the likelihood of these rats who have been pregnant before to show maternal behavior very, very high compared to rats who have never ever raised pups on their own or given birth. So there are other experiences that will impact maternal behavior. And one of those, a major influence, is just how much maternal care offspring received very early on in life. So again, remember, there are these differences between mothers. Some are good mothers and some are bad. There's just variation. And we can categorize them. So we can watch pups and see what kind of mother that they had. There are pups that are raised by good mothers and then pups that are raised by bad mothers. In this study, what they did is that they had those pups. They knew that they were raised by either good mothers, high LG, or bad mothers, low LG. And then they looked to see how much licking and grooming behavior these pups did once they were mothers. And they just monitored the frequency of licking and grooming over a period of time. That's what this is showing. And you can see that this is a significant difference. For those pups that were raised by a good mother, quote unquote, they show more licking and grooming behavior to their own pups when they are mothers. And that the low group, where they were raised by a bad mother, they show less. The pups that were raised by a good mother, they show a lot more licking and grooming behavior immediately after birth, postnatal. So this is showing the days of age of the pups. So 1 to 2 days old, 3 to 4 days old, all the way out to 9 to 10 days old. And you can see the biggest difference that we see initially is very early on. Over this first week, the frequency of giving licking and grooming behavior over a certain period of time is much higher in those pups that were raised by a good mother than those pups that were raised by a bad mother. And you can see that the difference starts to wane a little bit after about a week. But if you remember from the previous slide, there's only about 20 days that the mothers are raising the pups and providing lactation. After around 20-25 days, the pups are now kind of more on their own. So this is about half of like the mothering experience, and it's nearly twice as much. So it's a significant effect. So what about this experience then? Maybe this is something that's genetic, right? So maybe a high licking grooming mother is going to be more likely to give birth to high licking and grooming mothers. One way we can test this is to do cross fostering. And it turns out that it's actually probably much more influenced by the experience than the mother that actually gave birth to you. Because so one thing that's kind of nice about rats is that you can do this cross fostering experiment as long as you take the pups at a very early age and switch them. You can take a pup from one mother and give it to another mother. As long as they're about the same stage as far as raising pups, they gave birth at approximately the same age or the same time, you can switch them. You can flip them around. So in this experiment, this is simple, but it can be a little confusing to understand because we got two things going on here. So the black bars are pups that were raised by a high licking and grooming foster mother. So this is a mother that was not biologically related to them, but these pups were raised by these mothers. And then the open bars, the white bars, these are pups that were raised by a low licking and grooming foster mother. So these are the good mothers, and these are the bad mothers. Bad mother, good mother. Then we have two categories of pups, right? So we have pups that were born, so they were not raised by these mothers, but they were born from a high licking and grooming biological mother or a low licking grooming biological mother. So they were born to either a good mother or a bad mother. Now, if it was strictly genetic, you would expect then that when these pups are eventually have babies of their own, if it was strictly genetic, It wouldn't matter if they were raised by a high licking and grooming or low licking and grooming, a good mother or a bad mother, that they would show the same level of licking and grooming, that it would be really, really high. That is not what you see. Instead, pups that were raised by high licking and grooming mothers are more likely to show licking and grooming behavior when they have pups of their own. And it doesn't matter if they come from a high licking and grooming mother or a low licking and grooming mother as their genetic mother. Even pups that are born from a bad mother but raised by a good mother show very high percentage of licking and grooming behavior relative to those that were born from a high licking and grooming mother but raised by a low licking and grooming mother. So the data is pretty clear here that it really depends upon the mother that you're raised by and not so much the genetics. This also seems to have some. So what's the point of all this, right? What would be the influence of being raised by a high licking and grooming versus low licking and grooming? What difference does that make? So an experiment has been done like this. So we have pups that were either deprived of maternal care, that's the DEP, or non-deprived. And then we look to see what kind of response, what kind of cortisol response do they have. So we've got pups that are getting maternal care and pups that are not. This is the gray bar versus black bar. Then they were injected with ACTH and at different ages, postnatal day 4, postnatal day 8, postnatal day 12, postnatal day 16. Okay, so we're looking at a range of ages, and we're looking to see what is the cortisol response. So if you inject all the pups with the same amount of ACTH, then you can look to see how much cortisol do they release? How primed is their stress response? And you can see for every single age, for those pups that are already deprived, they have a much greater release of cortisol than those pups that are being getting maternal care. So this is an indication that maternal care reduces the stress response in pups. So all animals will be confronted with stress at various times, but for those that are raised by good mothers, or at least are getting maternal care, they have a more muted stress response. They have a stress response, right? There's cortisol that's being increased, but it's not nearly as much as it is for those animals that were deprived. And this seems to indicate that a poor mothering experience as an infant will lead to a much greater stress response and probably more likely to have negative outcomes than those pups that have that were raised by having good maternal care. What about some of the sensory stimuli? So briefly mentioned that there are all these sensory stimuli that will influence how the brain works. Let's walk through it in a little bit more detail. So we're going to go back to that previous experiment that we talked about with the induction of maternal behavior. This is called concavation. And so those previous experiments that showed the repeated exposure to rats that can induce maternal behavior, both in virgin females and females that had prior maternal experience. Females that have prior maternal experience, they're referred to as dams versus a naive virgin or nulliparous female. What is the point about the pup? Like, what is happening when you throw a pup in a cage with either type of female? It's inducing a change in these females, but what is it that they're sensing? Well, we know a fair amount about it. We know that pups emit vocalizations to elicit care. So if you take an isolated pup and you throw it into the cage of either a dam or a naive virgin, they're going to be hearing cries of that isolated pup. It's going to be whining. The pups, they exhibit a certain smell. So that each pup will have its own sort of smell, and it's a pup smell. And even humans, they'll talk about that new baby smell. That's a real thing. Warmth, of course, especially if the pups are healthy. This can induce care as well. So then the fact that the pups are kind of warm and they have, no hair on them, this is showing like what these little pups look like when you do these kinds of experiments. They're very, very tiny. They're little pink, kind of gross, but also kind of cute at the same time. The skin is very, very warm. So that's another thing that will elicit some of this behavior. Those are the things that the mother rats or the female rats are feeling. The pups, they also root around and they push against a rat's belly in order to elicit her to do what's called the nursing crouch reflex. So these pups, when they're hungry, they're constantly hungry. They constantly want to nurse, and they will, if the female rat is close to them, it doesn't even matter if it's the mother or not. These little baby pups, they will start to try to nurse, and they will push on the rat, and that will stimulate the rat to then show nursing behavior. And now all of a sudden you have nursing, suckling, inducing certain changes within the naive rat or the dam. So there's all kinds of factors happening here. So there's a lot of tactile, smell, some vision, plenty of auditory stimulation. So there's all these factors that are that the pup is emitting in order to induce the rat, the female rat, to show mothering behavior. Let's talk a little about some of the olfactory cues. So this is a very complex circuit. I do not expect you to have this memorized. We have talked about the circuit on the right. This is what we learned for the olfactory pathway. So we're revisiting some of the sensory inputs. This is why we have to cover sensory information first, because it's really the first thing that comes into the brain, and it will influence how we think about other things. So this is a very complex circuit. Again, do not have to have this memorized, but I want to illustrate for you that there's this additional pathway that has a huge influence over behavior. So we have the main olfactory epithelium, right? So this is where you have your olfactory receptor neurons with the cilia, and then they have axons that go through the cribriform plate and then go up to the main olfactory bulb. And in rodents, this is very, very big. It takes up a big part of their brains. And this is where you have the glomeruli, and then you have the mitral cells that output to a whole bunch of different brain areas, right? They don't go to thalamus, right? There's no connection to the thalamus directly. And instead, these mitral cells will synapse on a whole bunch of different areas, including the olfactory tubercle, piriform cortex, portions of the amygdala, entorhinal cortex. These are all olfactory input areas in this pathway. There's an additional pathway. We have, humans have this. It's very small in humans, and the degree to which it's important in humans isn't entirely clear. It does seem to have some influence over behaviors, but there's some debate about it too. But it's definitely well described in other animals, especially in rodents, because it plays such a big role. We have something called the accessory olfactory pathway. So there's another olfactory organ in the nose called the vomeral nasal epithelium. So this is a separate epithelium, and it's also referred to as the vomeral nasal organ. Humans have one too. It's really, really small. It's significantly bigger in rodents relative to the entire size, but it's still much smaller than the main olfactory epithelium. This vomeral nasal organ sends its projections to what's called the accessory olfactory bulb. Not the main olfactory bulb, the accessory olfactory bulb. So it's right up there near the main olfactory bulb, but this is just a secondary pathway. And then from projections from there are sent to different areas. They don't send projections to these cortical areas like the entorhinal cortex and piriform cortex, which is going to be very important for perception on the perception of how we perceive odors and recognizing odors. Instead, these will project to areas that are more involved with again, parts of the amygdala, so it's going to be regulating an emotional response, having projections then to the hypothalamus to induce hormonal changes. There's also the beta nucleus of the stria terminalis, the BST. This is known as a very important sort of instinctual behavior center. So you can see that there's just these additional areas. So the main point of this is that we have a secondary pathway that the smell of the infants, or say even from potential mates, this is being regulated through this pathway. So let's talk a little bit about how we know that that's the case. So this is involved with doing some lesions. Again, so pretty common type of experiment, and where you knock out the area and then look to see, well, how is this affecting behavior? And there's a very famous lesion study that showed how each of these regions play a role in controlling maternal behavior. And I'm going to walk through this. It's a little bit counterintuitive, the results, but it sort of makes sense. And I'll explain why it makes sense at the end, but let me walk through the results first. So in this experiment, they either lesioned the olfactory bulb, the OB, or they lesioned the vomeral nasal organ, or both together, VNOB both of them being lesioned. They had some that received the vomeronasal lesion that got some of the olfactory bulb as well. So they just refer to these as VN+. When you're doing these kinds of lesions, especially in a rodent brain, everything is really small, and all these things are pretty close to each other. Sometimes you're trying to hit one area, but when you look after, in the brain to make sure that you hit the right area, sometimes you get that area plus a little bit extra. That's what the VN+ is. So we got the vomeronasal organ lesions, olfactory bulb lesions, both, and then VN+. We also have sham, right? So sham means that these animals were anesthetized, that you put in, say, an electrode to try to burn out the area, but you didn't actually burn out the area. You just did everything except lesion the area. That's just to control for doing the surgery, because that could mess them up as well. So they did this, and then each female was presented with foster pups. So we're doing this foster concavation study where we were trying to induce maternal behavior. And normally, and that's what we're seeing with the sham, is that for these naive females, it takes some time for them in order to induce a maternal behavior. It takes like 6 to 8 days in this study. It took a little bit of time, but eventually they start to show maternal behavior. And so, right, that's the control. When you lesion the olfactory bulb, you have a little bit of reduction, but not a significant decrease in the reduction of the amount of time. So animals that are getting this lesion are now a little bit more likely to show maternal behavior. When you do the vomeronasal organ just by itself, now this is a significant difference. This is the time at which it takes to induce maternal behavior is shortened. It's around 4 days versus more than 8 days. When you do VN+, getting both of them, it's now like even less than 3 days. And when you get both, this is when you get the maximum response. You eliminate both the vomeronasal organ and the olfactory bulb. Now these naive females are much more likely to show maternal behavior. This seems counterintuitive. You would think you need this system in order to show the maternal behavior, but instead the system of recognizing odors appears to be inhibiting showing the maternal behavior. This is showing that the smells that are involved here are actually an inhibitory response. These females are not primed to show maternal behavior yet, right? They are nulliparous. They haven't gone through this experience of having the surge in oxytocin. Having an intact olfactory system is part of that. These animals use smell as a way of recognizing whether infants are truly theirs or not. And eventually the mother will, the nulliparous female will eventually just be overly influenced by all the additional stimuli, and it will counter the smell portion of inhibiting giving maternal behavior. But if you eliminate this olfactory ability, now the mothers are more likely to show olfactory behavior because they do not have the ability to distinguish whether this new foster infant is truly theirs or not. So, and this makes sense. These mothers, they're not mothers. These are nulliparous females. They do not want to be raising pups that are not theirs. They have not undergone a pregnancy. They have not been primed to think like, oh, I should take care of babies. And rats are relatively social creatures. If you are a female who has never been pregnant or given birth to babies, there's no point in you taking care of someone else's babies. So this mechanism of smell and smell recognition is one way of ensuring that that a nulliparous female isn't being tricked into taking care of infants that are not theirs. But if you eliminate this smell system, they can't recognize kin from non-kin, and now all of a sudden they're more likely to give maternal behavior to infants because the other stimuli, the whining and the presence of the smell, sorry, not the smell, but the touch and the warmth, all of those factors will induce them to engage in mothering behavior because they don't necessarily smell that it's not theirs. Does that make sense? That's the interpretation from this. It's a little counterintuitive that by destroying parts of the brain that you're more likely to see a behavior, but it's clear that smell is important in preventing females who have never given birth from taking care of babies that are not theirs. The last thing we're going to discuss, and we've already touched on it a bunch, is hormones and their role in inducing maternal behavior. There are a bunch of hormones that obviously are involved: estrogen, progesterone, and prolactin. We're not really going to discuss prolactin at all, but estrogen and progesterone, we'll talk a little bit more when we talk about sex. Instead, we're going to focus, again, just on oxytocin. So hormones that affect maternal behavior, such as oxytocin, we're going to look at treating virgin females with oxytocin to see what happens to their overall maternal behavior. So in this case, we have rats that are getting either oxytocin or they're just getting a control substance. In this study, they call it EB only. So we've got oxytocin versus EB. The oxytocin is the darker gray bars here, and EB is just the controls. And then we have maternal scores. So we're categorizing, how maternal these rats are. These are virgin females, so they've not given birth, but we're just injecting them with oxytocin and then seeing how likely they are to show maternal behavior to orphan pups. Remember, it takes days and days and days typically for a virgin female that has not had its olfactory system destroyed to show maternal behaviors. If you inject them with oxytocin, you're more likely to get more maternal behaviors. So 4s and 5s, these are rats that are showing quite a few maternal behaviors, versus 1, 2, or 3 is somewhere in the middle. You can see it's hardly any of them, versus 0, which is showing no maternal behavior. So animals that are controls, you can see that more than 60% of the group shows no maternal behavior or very little. And it's around like 20% of the rats, they show some maternal behavior. It's not anything close to what you might see in a rat that has undergone birth, but it's some maternal behavior. If you just inject a naive rat with oxytocin, you can see you can cut the percentage of those rats that are showing no maternal behavior to less than half, and that there's more than 60% of the rats are showing maternal behavior. This is all within an hour. So an hour of being treated, they are much more likely to show maternal behavior. Clearly, oxytocin is playing a major role in inducing these changes. This is another experiment showing the effects that it has on this concavation experiment, where you inject animals with oxytocin into the brain. So they're injecting this directly either into the brain with oxytocin or just a control substance. And then they're looking to see whether they're more likely to show maternal behavior or not. And then we're looking at the cumulative changes over days of pup contact. So this idea that, with controls, normally it takes about 8 days for the pups for the naive females to show maternal behavior, right? Over time, you're going to see more and more females showing maternal behavior, and eventually they will all be doing it after about a week or so. Animals that were injected with oxytocin into the brain, and they're getting these daily injections, they are more likely to show maternal behavior very early on. You can see on the very first day, it's around 60%. 2 days of pup contact, it's more like 75%. 4 days, it's nearly over 80%. And by day 6, 100% of the animals are all showing maternal behavior versus only about 65% in the control animals. So this is oxytocin can induce the mothering behavior. It can also speed up and increase the likelihood of showing mother behavior plus pup contact. Another experiment that's been done is to block. So this is showing, the previous experiments show that oxytocin is sufficient to induce mothering behavior. These are experiments that are going to show that it's necessary to induce mothering behavior, right? So just giving them oxytocin, you can induce mothering behavior. What happens if you block oxytocin? So if you block oxytocin by injecting oxytocin antagonists, these are chemicals that block how oxytocin acts in the brain, you can lead to changes in mothering behavior. So we're going to look at 4 different kinds of mothering behaviors in animals that were injected with either saline. And these are not naive rats. These are rats that are primiparous. So they had already given birth once, and then they're given pups afterwards. Okay, so this is different than the than the naive rats. These are actually rats that are very likely to show mothering behavior when given an orphan. So we have rats. Some of these rats were injected with saline, and other rats were given this antagonist, OTRA. What you can see, so let's just go through the different experiments. In the first one, we see how long it takes for them to retrieve the first pup. So you put a pup in, you see how long it takes for them to go and retrieve the pup, numbers of seconds. And for a rat who has already given birth once, they retrieve that pup right away. There's hardly any delay whatsoever. But if these rats have been injected with the antagonist, you can see that now there's a huge delay in how long it takes for the females to go retrieve the pup. So it's inhibiting their ability to go retrieve a pup. You can look at the total number of pups received. So like in this experiment, I think they put 10 pups in the cage and they looked to see how many pups they actually retrieve. For the animals injected with saline, they retrieve nearly all 10. Whereas for those injected with the antagonist, they retrieve many fewer pups. Time spent in directed behaviors. So this is like the mothering, licking, and grooming behavior. So the saline treated pups, the ones that are injected with control, they show a lot more mothering behavior than those injected with the antagonist. And then the last thing is time spent exploring. So this is time spent away from the pups. Like when you're a busy mother, you got to take care of your babies. And if you're not that great of a mother, you're not going to be spending as much time with your babies. Rats that were injected with the antagonist spend more time exploring in non-maternal behaviors like exploring the cage compared to the rats that were not injected with the antagonist. So this experiment shows that not only do we need to have oxytocin, it has to be there working in order for this to work. So for these rats, it shows that oxytocin is also necessary. So by blocking the oxytocin, we are inhibiting their ability to show normal maternal behavior. So oxytocin can induce it, and it's also necessary. Another way to show this is to use genetic knockouts. So that's one thing that's nice about mice, especially, that if you for mice that have a genetic deletion in one of the oxytocin receptors, they will be deficient in showing maternal care. So this is looking at the percent of pup abandonment. Sometimes mice will show they will kind of give up their babies and no longer take care of them for a variety of reasons. Stress sometimes can induce this kind of thing. So we have mice that are wild type and they have normal oxytocin receptor expression. And then we have other mice that were knockouts. So they do not have a functioning oxytocin receptor. So even if they have a surge in oxytocin, it won't make a difference. It can't act on the brain. And these mice are much more likely to show pup abandonment. It's more than 60% compared to around 20%. It looks like oxytocin is also involved with social behavior, and that and this is true in humans, it looks like. So there's some genetic variability in the oxytocin receptor within humans. There's some that have either in the nucleotides, there's a small little difference, some variability. We see some that have AA or AG as a variant, or they have GG. And it looks like the ones that are GG tend to show more empathy. So they looked at humans, they assessed what kind of variation of oxytocin receptor they have, and the ones that are, and then they ran them through a series of tests looking at empathy and startle response. So like how stress response that they show given a certain challenge. So humans that have a GG variation in their oxytocin receptor are more likely to show empathy. So they have a higher empathy response accuracy in a test. This is true for males especially. For females, it's a less of a difference, but for males, it appears to be a significant difference. And but for both males and females, it appears that the GG variant has a lower startle anticipation heart rate. So their heart rate is a little bit lower when they're put into a startle response. So their overall stress response is less. So not only does prior experience seem to have an influence, hormones clearly play an experience, have an influence as well. But um, hormones also need to work with the underlying genetic material, and there appear to be certain behavioral phenotypes that correlate with differences in overall empathy output and stress response. Uh, it's it's not huge differences. You got to look at the scale here, right? Percent difference. It's between, um, a little less than 75% to a little bit more than 80%. So it's not a huge difference, but it's a significant difference. Now that we've covered oxytocin, I've spent a lot of time talking about it. Now we're going to cover a little bit about vasopressin, and we'll revisit oxytocin a little bit near the end. But let's first just talk about vasopressin. So vasopressin, as I mentioned, has this important peripheral role that are to regulate water retention, acting on the kidney, as well as blood vessel constriction. So water deprivation in the body, it decreases the overall body volume and increases salt concentration. There are peripheral sensors that send this information to the hypothalamus. The hypothalamus will then release vasopressin. This is the abbreviation for vasopressin, antidiuretic hormone. Then vasopressin acts on the kidneys to retain water and reduce urine production. So this process of when you are a little bit too thirsty, it's vasopressin that's playing a big role in that. That is one of the main roles of vasopressin. So then you have less water lost in urine, this normalized blood water levels, and then your salt concentration returns to what it should. Vasopressin, it looks like it has an additional role on acting in the brain. It seems to influence certain types of behavior, in particular social bonding. A lot of this work has been done with a very interesting and somewhat unique animal model called the vole. So here's a picture of what a vole looks like. They are a rodent, clearly. They look a lot like a little mouse, almost like a cross between a mouse and a gerbil. And but they have some really interesting social structures that make them valuable animal models for research. We're going to walk through a series of experiments done with one species of vole called prairie voles. It's shown right here, prairie voles. The thing that's really interesting about prairie voles is that they will show a partner preference. This is somewhat unusual amongst rodents. Rodents will mate, and then typically they don't care about each other post-mating. Prairie voles are different. They will show affiliative behavior. They'll want to spend time with their mate and show a partner preference after they have mated. Oxytocin appears to mediate this preference in females. So let's walk through this experiment a little bit. In this experiment, they did a couple of things. They have a female spend time with one male, either a male that she mated with or a male that she did not mate with. And so we have the dark bars. So she's spending time with a male that she did not mate with versus time with a male that she did mate with, okay? They infused certain drugs into the brain. So let's talk about this first part first. So they either infused a control substance or oxytocin into the unmated female's brain when she was with one of the males. Then they put her in a cage where she had to choose between two males, and then they looked to see how much time she wanted to spend with the male that she was either with when she was infused with control, or females that were infused with the oxytocin. Females that got oxytocin, they spend 90% of their time with the male in which they were exposed to the oxytocin. In a separate experiment, they had used mated females. So these females had already mated with one of the males, and then they looked to see how much time they spend with that male. And you can see in the control females, they are much more likely to show time with their mated partner versus the unmated partner. But if you infuse an oxytocin receptor, OTA in this case, into the female brain, it blocks partner preference. So even though she has mated with this male, when you infuse this oxytocin receptor, she no longer shows preference for the partner. Instead, in fact, it almost looks like she's spending more time with the other partner. When you do this experiment in males, it didn't have any effect. So this seems to be a female-specific behavior, and it seems like oxytocin is regulating this process in prairie voles. Vasopressin seems to be the mediator of partner preference in male voles. So when you infuse arginine vasopressin it's essentially vasopressin into an unmated male brain, he is more likely to show preference for the female that he was with when he got the vasopressin. When you block a vasopressin antagonist when you put a vasopressin antagonist into the brain to block vasopressin reaction, now that male will no longer want to show partner preference for the female that he actually mated with. So normally he would want to spend time with his female partner, nearly 80% of his time. But if you put an antagonist in there, you can break that bond. And if you do these kinds of experiments in females, it seems to have no effect. So depending upon the sex of the animal, it seems that oxytocin plays a big role in this partner preference in females, but it's in fact vasopressin that seems to regulate this partner preference in males. So we're going to talk a little bit more about that. This is what that partner preference looks like in the previous experiment. And so you'll put your focus animal, your experimental animal, in a cage, and then they will have a partner on one side or a stranger on the other side. And then you look to see whether they're going to spend time with one or the other. There are two species of voles here that show differences. So the prairie vole is monogamous. That's the one we just talked about with the oxytocin and vasopressin. And then we have the montane vole, which is promiscuous. So if you do an experiment to look just to see these species, so this is great because now you have two species that are closely related. They show very different kinds of social preference, partner preference. One way to keep this in mind is just to think that it's opposite. Prairie voles are monogamous and montane voles are promiscuous. So M goes with P, P goes with N. You can see the prairie vole wants to spend time with its partner versus alone or with a control. Montane voles, they want to just chill by themselves. You can see that in the montane vole, they want to spend more time just by themselves than with either their partner or with a stranger. So what's the difference between these two species? So a lot of this work had been done by Thomas Enzel, who was the head of the National Institutes of Mental Health and the National Institutes of Science, National Institutes of Health, the NIH. A lot of this work when he was in Thomas Enzel's lab was done by Larry Young, who's a major professor at Emory, one of the top professors or scientists working on partner preference and neurobiology of regulating partner preference. So what Larry's work shows is that there's a big difference between the receptor expression between montane voles and prairie voles. So it's not so much about the hormones themselves as it is about the receptors. So they have this distinct overall mating behavior, but it looks like prairie voles have a huge number of receptors, a lot of high receptor expression in many different brain areas. So this is just showing the in situ hybridization for the oxytocin receptor in prairie voles versus montane voles. And then in males, you can see that this portion of the brain actually has a very in the more ventral portions of the near towards the hypothalamus. It's called the dorsal band or the diagonal band. I'm sorry. That there's really high expression of the vasopressin receptor V1A in this area in prairie voles versus montane voles. And there appears to be, and this is just a summary of this difference that with the prairie vole, they're monogamous. Both parents take care of the babies, and there's a lot of high partner preference and selective aggression. Montane voles seems to show very opposite behaviors, a lot more sort of typical mammalian behavior, more promiscuous. It's just mothers who take care of the babies, and they show very low partner preference. So is this the difference? The fact that you have this very stark difference in receptor expression? Well, Larry actually tested this. What he did is that he injected a virus into the brains of montane voles that overexpressed the VA1 receptor in this diagonal band that's down towards the ventral portion of the brain in the hypothalamus. So now he's now made mice that are montane, sorry, not mice. He's made voles, these montane voles, to look a lot like prairie voles by injecting this receptor. So he injects this virus with the receptor. Now all of a sudden they're expressing a lot more of the receptor in these same brain areas versus a control virus. So this is a virus, you got to do the controls right. So you got to do the surgery, inject them with a virus. You want to make sure it's not just because you injected a virus into the brain of the vole. But there's no increase in V1A receptor here. So doing the same in situ hybridization, you don't see a difference. Instead, it's just the virus that is expressing the V1A receptor. So you're turning the brains of these male montane voles to make them look like prairie voles by making them express more receptor. And you can see that in these montane voles, they spend a lot more time with their partner when they have been injected with the vasopressin receptor. So you can make a montane vole act like a male prairie vole by forcing their parts of the brains that express this receptor, the vasopressin receptor, to make the receptor. So we're not going to spend a lot of time talking about aggressive behavior, but it is a form of social behavior. And mice are great for looking at aggressive behavior because they tend to be pretty aggressive. Male mice will fight with other male mice. And there's some evidence that indicates that vasopressin plays a role in facilitating aggressive behavior in mice. So we have two mice here, one that is wild type, that's what the ++ means, and we have another mice that has a mutation in one of the vasopressin receptors. So this is the 1B version of the vasopressin receptor. And so it's minus, they're mutants. Then they look to see how many defensive attacks they show in various tests. And they did I don't remember the details of the test, but it's like an increasing scale of the kinds of tests. They did 3 different tests, and then they looked at the combination of them. And you can see a mouse line. One of the mouse lines that had the mutation showed hardly any defensive attacks. So they showed no aggressive behavior in 2 of the 3 tests, and fewer defensive attacks in the last test. So clearly a huge difference in the percent of showing defensive attacks over time. And so clearly they need a working vasopressin receptor in order to show normal aggressive responses. When the receptor is genetically added to the hippocampus, they can now restore aggressive behavior. So it appears that the hippocampus is part of this. So here we have the wild type. The percent of mice that are attacking, it's around 50% in this particular test. The knockout shows, so it's around 10% of the mice are showing the aggressive behavior. And then if you replace the receptor just into the hippocampus, you don't get a full recovery, but you get a much greater increase of the percent of mice that are showing aggressive behavior versus the knockout. So this is knockout with virus that allows them to express the behavior. So I just wanted to touch on the interactions of vasopressin and oxytocin and its overall and its relationship to aggressiveness in dogs. So dogs with a history of aggressive behavior, those are shown as cases, they have higher levels of circulating vasopressin than those dogs with no history of aggression. So more aggressive dogs have higher circulating vasopressin. Is it acting on these aggressive brain areas to induce them to have more aggressive-like behavior? That's what this data would suggest. This correlation, but it's consistent with that interpretation of the experiments that we've seen in mice. In this same study, they also looked at oxytocin levels. And in this case, instead of looking at aggressive dogs, they looked at dogs that have been selected for being assistance dogs. So these are therapy dogs. They're picked because they are super nice, they're sweet, and they love people, and they're not going to show any aggressive behavior. These dogs actually tend to have higher levels of circulating oxytocin versus dogs that have not been trained to be assistance dogs. So, are they picking dogs to be assistance dogs because they're more likely to have higher levels of oxytocin, or is the training of being a caring dog leading to them to have more oxytocin? That's a great question, but this study is just correlational, but it shows a significant difference between the two. So in conclusion, there's a lot that we've discussed today focused on vasopressin and oxytocin, and so there's a series of questions to focus on for key questions related to this presentation. So this all started with the posterior pituitary, the neurohypophysis. So you should know how it differs from the anterior pituitary. What are the functions that oxytocin regulates? How does maternal behavior look in rats, and how does it change with experience? So understanding all of the naive versus primiparous rats and how they respond to orphan pups. What are the effects of manipulating oxytocin signaling on maternal behavior? All those experiments where you inject oxytocin into the brain or inject oxytocin receptors. What are the functions of vasopressin? A little bit, we focus mostly on the social behavior aspect of it. What are the major differences between prairie voles and montane voles in their brains and behavior? So this is one question that summarizes basically all of those experiments that we talked about. And then what does the manipulation of vasopressin do to courtship of behavior in male voles? Lastly, what is the role of vasopressin in aggressive behavior in species of mammals? That's it. We'll talk to you later.